screen here. All right. Can everyone hear me okay?
Yep, you're all good.
All right. All right, so thank you for the introduction. So today's talk is going to focus on cellular energy transformations that happen in our magnificent mitochondria. Before we get into the focus of our talk today, I'm just going to tell you a little bit more about the course that I teach in. So the material that I'm presenting today is coming from Biology 241. And this is the biology course that we offer in the fall semester. And this course is centered around the idea of energy flow in biological systems. So I teach in this course and I also help coordinate the course. I'm going to be helping to coordinate the labs in this upcoming fall semester. So at the end of the presentation, if you have any general questions about biology, about labs, about studying in biological sciences, I'm happy to answer any of those kind of questions. But first of all, I just want to tell you a little bit about what this course covers. So we're focused on energy transformations, and we start small. So we start at the molecular scale, and we're thinking about the energy transformations that happen at this level. So the video that we're seeing here is a protein called kinesin in pink. And this protein looks like it's walking along this microtubule that's present inside a cell. Attached to the kinesin is this package of cargo. So what's happening here is really an energy transformation. This protein is taking our universal energy currency inside our cell, called ATP, and it's converting the energy in that ATP into kinetic energy, so into movement along the microtubule. So in the course, we start small with molecules, with proteins, and then we move up in scale. So then next we look at cellular energy transformations. This image here is a eukaryotic cell. We see the nucleus in the middle, some organelles, membrane-bound organelles within the cell. So one example of a cellular energy transformation is cellular respiration. So taking the chemical energy in the food we eat and converting it into ATP so that our cells can do work. So that work could be transportation of cargo. If I open the chat here, feel free to pop in any other ideas. So what other work might a cell do that requires energy? Anyone want to offer some ideas in the chat here? So other examples of work that a cell might do, so they might replicate their genome, they might synthesize new biomolecules, new proteins. So all of this work that a cell does is going to require energy. Okay, so then moving up in scale, the second half of the semester is going to focus on organisms and ecosystems. So here we have hummingbirds feeding nectar. So energy has to be used in a very strategic way by organisms. So they need to be able to allocate that energy for different processes like thermal regulation or for reproduction, anything locomotion. We see the hummingbirds here flying. So that energy has to be allocated for each aspect of the organism's life. And then finally, we talk about energy flow in ecosystems. Okay, so this is a picture from Sunshine Meadows by Banff in Alberta. So this is an example of an ecosystem. In the course, we study energy flow within that ecosystem, and we look at connections between our primary producers, so the plants that are growing here, our primary consumers, so the animals that feed off those plants. It could be rabbits, it could be deer, and then the secondary, tertiary consumers like the grizzly bears, wolves. So we look at the energy flow within the food web, and then also nutrient cycling within that environment. So this is an overview of what the course is about. And in today's sample lecture, I'm going to share a snapshot of some of the topics that we cover that have to do with cellular energy transformations. So really thinking about mitochondria and their role in these energy transformations. But before we dive in there, we're going to back up and actually think about the question, where do mitochondria come from? So in order to answer that question, we're going to dive back in time. So thinking about towards the 1700s, so scientists like Carl Linnaeus started questioning how to organize the life that we see around us. So beginning in the 1700s, scientists typically classified organisms based on morphology or how they looked. And this system was used and refined for hundreds of years. And then around 50 years ago, so around the 1970s, there was this molecular biology revolution. So scientists were beginning to understand the molecular details of how we go from DNA, the sequence of DNA, to RNA, to protein. So they were beginning to understand that flow of information. And they were also beginning to develop tools to study that process and manipulate it. So they were able to look at sequences of DNA within organisms. So around this time in the 1970s, a scientist named Carl Woese wanted to study organisms that were living in really extreme environments, like the hot springs in Yellowstone. So this classical organization system of morphology becomes very difficult to apply when you're working with microscopic organisms and organisms that you can't easily bring into a laboratory setting and grow there. So Carl Woese and other scientists wanted to ask the question, what lives here? And with the new molecular biology techniques that were being developed, they were able to answer that question by studying the DNA sequences inside the organisms that lived in these extreme environments. So by sequencing DNA from these organisms and comparing those DNA sequences to other organisms, they proposed a new classification system of organisms that was based on genetic information. So they were able to classify all life into 3 domains. If we start at the bottom here, we have domain bacteria, then above domain archaea. So archaea primarily live in these extreme environments like the hot springs or areas with really high salt. And you'll notice that both domain archaea and bacteria are prokaryotes. So these are prokaryotic cells, meaning that they lack a nucleus. The third domain that we see here is domain eukarya. So domain eukarya includes plants, animal, fungi, as well as unicellular eukaryotic organisms. And to distinguish them from prokaryotics, these are organisms that have a nucleus, so the membrane enclosure around their genetic material, so around their genome. On the right-hand side of the slide, we have a stylized phylogenetic tree where we see the 3 domains of life, bacteria in blue, archaea in purple, and then eukarya in orange and green. And what you can see from this phylogenetic tree is that these 3 domains do not have entirely independent evolutionary histories. And what I mean by that is that we have these crossover events that we call horizontal gene transfers that occur between domains. So thinking about that idea, that horizontal gene transfer, I have a question here for you, a Menti question. So you can join Menti here using the QR code, or you can join at menti.com and use this code at the top. So thinking about this, I have 3 scenarios below, and what I want you to do is think about which of these scenarios would be an example of a horizontal gene transfer. So this is a bit of a guess if you're not familiar with that terminology, but I'm going to use it to explain. So 3 scenarios: a bacterium divides into 2 identical daughter cells. A bacterium called Shigella transfers a gene encoding a toxin to a different bacterium called E. coli. Third scenario, an organism passes a genetic mutation to their offspring. Okay. I'll give you a few more seconds to join in if you want to try the Menti question. Thanks to those who've submitted an answer here. All right, so the correct answer here is the one that the most people chose. So we have 9 now. So the transfer of a gene from one organism to a different organism. So we have a gene moving from one species to another. So that is the definition of horizontal gene transfer. The other two scenarios are referred to as vertical gene transfer, where an organism transfers genes to its progeny or offspring. Okay, so horizontal gene transfer is happening between different organisms. Okay, so if I go back to our presentation here, we can see that's what's happening between domain bacteria and eukarya here. So we have this arrow here, bacteria that gave rise to mitochondria, and that is a horizontal gene transfer event. So we're going to take a look at this horizontal gene transfer, which is thought to be a consequence of an endosymbiotic event that happened over 1 billion years ago. So this process is described in the endosymbiotic theory. So if we think about the word endosymbiotic, the term endo means within, and symbiotic is living together. So living together within. Okay, so if we start at the time scale here, you can see around 3 to 5 billion years ago, life was prokaryotic. And then starting around 1.5 billion years ago, we see that we begin to have unicellular eukaryotic cells. So we're starting here 1.5 billion years ago, life is prokaryotic, and we have this original prokaryotic host cell. Then we have this endosymbiotic event. So these aerobic bacteria begin living within another cell. We see they're entering the cell, and this association or symbiotic event is beneficial for both organisms. So when the aerobic bacteria are living within the host cell, they're protected from the harsh external environment. And when the host cell has these bacteria living within it, these aerobic bacteria are really good at producing energy, and the host cell can use that energy. So they have this association that's beneficial for both. And over time, as they live together, they become dependent on each other and they can no longer live apart. So those aerobic bacteria are no longer able to be removed from that cell and live independently. So over time, the bacteria became mitochondria, and the host cell evolved into an early eukaryotic cell. So here we have the mitochondria. Over time, the cell is developing a nucleus here. It's turning into a eukaryotic cell. So our eukaryotic cells, so we have our unicellular eukaryotic cells. We can also then have animals, fungi, and some protists as well. Okay, so and also, going back to that idea of horizontal gene transfer, so exchange of genetic material from one organism to another, not from one organism to its offspring. In this case, we have horizontal gene transfer because some of the genetic material that was present in the original aerobic bacteria actually moved into the nuclear genome of the host cell. So the mitochondria itself actually still has some genetic material within it, so it has its own genome, but the majority of genes that were once here have now been transferred into the nucleus of the host cell. So that's that horizontal gene transfer event. And there's a few pieces of evidence that support this theory. So one is the size of the mitochondria is very similar to modern day bacteria. The mitochondria actually divide like a bacteria does, so they both divide with binary fission. And the mitochondria still retain that genetic information. So they have their own genome, they have genes that encode for some of their own machinery. But you can no longer take the mitochondria out of the cell and have it survive and replicate on its own. So they're dependent on each other. All right, so now we're going to take a closer look at the mitochondria. So we saw where, or this predominant theory about where they came from. Let's take a look at the structure here. So we'll see right away that the mitochondria actually have this double membrane. So they have an outer mitochondrial membrane that we see here. They have this inner membrane that has all of these folds in it. And then we have two spaces to think about. We have an intermembrane compartment between that double membrane. As well, we have a matrix, which is inside both membranes. Okay, I have one more Menti question for you. You guys did really well on the last one. Might feel like I'm jumping a little bit here, but this is going to introduce the next idea. So which cell is smallest in diameter? So we have three cell types here. Adipocyte. So adipocyte is, we have them in our body. They're used for storage of fat. E. coli, which is a bacterium. And then yeast, which is a unicellular eukaryotic organism. Okay, I'll give you a few seconds there to join if you want to answer this Menti question. Oh, so far we're split. Nobody thinks one for adipocyte. Any other guesses? Oh, okay, E. coli is winning. Another one for adipocyte. All right, so I'll share the answer with you now. So out of these three cell types, the adipocyte is actually the largest. So they can range in diameter from 20 to 300 micrometers. The second largest is the yeast cell. So the yeast has a diameter of about 5 micrometers. E. coli is the smallest. So E. coli, and in general, most bacteria have a diameter around 1 micrometer. So our smallest cell here is E. coli, which is our prokaryotic cell. And the reason why I'm asking you that is that we're going to think about mitochondria in the context of cell size. So in this diagram, we're comparing two types of cells. A eukaryotic cell on the left, you can see we have our nucleus in the center here. On the right-hand side, we have our prokaryotic cell. The cell is more simple in terms of its organization. It lacks the nucleus. It has its genetic material here. As they're drawn, they look to be the same size until you check out the scale bar at the bottom. So 1 micrometer scale bar for our bacteria, our prokaryotic cell, 10 micrometer for our eukaryotic cell. Okay, so our eukaryotic cells in general are about 10 to 100 times larger than a prokaryotic cell. So we can ask the question, how are eukaryotes large? We can also ask, why are prokaryotic cells small? So this is kind of the limit of how big a prokaryotic cell can get. There's always exceptions, but in general, prokaryotic cells, their diameter is around 1 micrometer. What prevents them from growing larger? The answer has to do with surface area and the internal volume of that cell. So in the prokaryotic cell, it has the surface area, which is its cellular membrane, and it has its internal volume here. And it needs a certain ratio of surface area to volume in order to support the needs of the cell. So at this plasma membrane in the prokaryotic cell is where all the ATP is generated that needs to support this volume. The surface area also allows for passage of molecules in and out of the cell. So we need an adequate amount of surface area in order to support the volume. Now imagine for a second that you're taking a balloon, you're blowing it up. As that balloon is expanding, the internal volume actually increases at a greater rate than the surface area of the balloon. So the same is true when we think about cell size. As we increase the size of our cell, the volume within that cell increases at a greater rate than the surface area. And that's the reason why the prokaryotic cell needs to be small. It needs to have enough surface area to support the inside. If it gets larger, then that surface area to volume ratio is no longer favorable, and that surface area isn't able to support the internal volume. So now we can think about the opposite question: how can eukaryotic cells be large? And the answer to that connects back to mitochondria. So eukaryotic cells 10 to 100 times larger than the prokaryotic cell. That means if we think about surface area only as the plasma membrane here, it's not enough to support the internal volume of the cell. However, the eukaryotic cell has hundreds of mitochondria within it, and the mitochondria have lots of surface area, and that's the site where ATP is generated in the eukaryotic cell. So this is what allows that eukaryotic cell to grow large. Now, in the next slide, we're going to take a look at mitochondria as these metabolic powerhouses. So they're the organelles of the eukaryotic cell that are generating this cellular energy currency, ATP. So when we think about generation of ATP, we're talking about cellular respiration. So the energy from the food that we eat is transformed into ATP. When we think about this process, we often talk about oxidation of glucose. So in this diagram here, on the y-axis, we have free energy. On the x-axis here, we have progress of a reaction. Okay, and when we think about oxidation of glucose, this can happen just by reaction of glucose with oxygen to form carbon dioxide and water. And so we can see here on this reaction, so these are our reactants, our products. We have this activation energy barrier that we need to overcome in order to get this reaction going. So if this reaction is happening outside of a cell, we can just use a flame. We can heat up the glucose in the presence of oxygen. So we are using combustion, and we will form carbon dioxide and water. But in this process, all of the free energy present in glucose is going to be released into the surroundings as heat. And so that heat isn't going to be very useful to do work. Again, remember, in the cell, we want to use the energy from our food to do work. So how does this process look in a cell? So if we move over here, we're looking at oxidation of glucose, but by cellular respiration. We also can't just light our cells on fire. We obviously can't withstand that heat to overcome the activation energy. So instead, this process is divided into a series of small steps, each with a small activation energy that can be overcome in the cell. We also don't want to create a ton of heat that's not useful for doing work. So instead, the energy released through each of these reactions that occur during cellular respiration is going to be transformed into a different type of energy. So this is where we start to get into these energy transformations. So instead, the energy that was present in glucose initially is going to be transferred to special carrier molecules in our cell. So what's happening is that high energy electrons are transferred from glucose onto these carrier molecules called NADH and FADH2. Okay, so if we think about where this is happening, so here's our eukaryotic cell. We have our cytosol, so this is the fluid within the cell in orange here. So during cellular respiration, the energy that's from our food is going to enter the cell. And when we talk about this process in class, we're really focusing on carbohydrates, although the energy can come from fats and proteins as well. We focus on carbohydrates, and we focus on that energy coming from the building block of carbohydrate, glucose. So that glucose enters our cell, and through the process of glycolysis, it's split. Some of the energy is transferred. Some of it's used to make ATP, but just a tiny bit of ATP. The rest of the energy is present on our carrier molecule, NADH. And some of it is present on pyruvate, so that's the carbon-based product of glycolysis. Pyruvate then enters into the mitochondria, so the rest of the cellular respiration process is happening in the mitochondria. And by oxidation of pyruvate, we produce more of our carrier molecule, NADH. We have a molecule called acetyl-CoA that enters the citric acid cycle. It's further oxidized into carbon dioxide. So this is what we're exhaling. And the energy that was present initially, we have a little bit of it forming ATP, but the majority of that energy, again, is in these high energy electrons that are present on NADH and FADH2. So we've transformed our energy from glucose onto these electron carriers. The last step is to really make the bulk of the ATP. And this happens through a process called oxidative phosphorylation. And this is the process that actually requires the molecular oxygen that we're breathing in. So on the next slide, we're going to take a closer look at oxidative phosphorylation and the energy transformation that's happening in the mitochondria. So just as a reminder, most of the energy that was present on glucose is now present. We've made small amounts of ATP. The majority of the energy is now on these high energy electron carriers, NADH, FADH2. Okay, so we're moving into the mitochondria. So just a reminder of its structure here, we have our outer membrane, we have our inner mitochondrial matrix. There's a space between those two membranes called the intermembrane compartment, and then the matrix in the middle. So going to the left-hand side here, just to situate you, this is our inner mitochondrial membrane. Our matrix is down below. Intermembrane compartment is above here. So through the series of oxidation reactions that have happened in our mitochondria, we now have lots of these high energy electron carriers, NADH and FADH2. So we need to take the energy from these molecules and use it to make ATP. And the way that the mitochondria does this is through a series of complex energy transformations. And it's a very, very elegant way to make ATP. So these high energy electron carriers arrive at a series of protein complexes at the inner mitochondrial membrane that we call the electron transport chain. And then the electrons are passed through these protein complexes and also some mobile electron carriers until they finally reach oxygen. So this is the oxygen that we're breathing in that we need for survival. When the electrons flow through the electron transport chain, through the series of redox reactions to oxygen, then we form water. So this was our other product of the reaction. And because these series of electron transfers are a spontaneous process that releases free energy, that free energy is used to create an electrochemical gradient. So these components of the electron transport chain are using that energy from the electrons to pump protons into the intermembrane compartment. These protons are not able to diffuse back through the membrane. So this membrane is really hydrophobic. It hates water. It hates charges. These charged protons can't move back through. So we've created an electrochemical gradient where we have lots and lots of protons that have this charge associated with them. Okay, and the next part, I'm going to introduce an analogy here of a hydroelectric dam because the process is very similar. So in a hydroelectric dam, we have this dam. So we have a big pool of water. When we open the dam, the water is going to flow through until it reaches a turbine. Then it's going to spin the turbine. And that kinetic movement of the turbine spinning, the generator converts that energy into electricity. So a similar process is happening here in our mitochondria. So we have this gradient of protons. It can't move freely through the membrane, but it is able to move through this molecular machine called ATP synthase. This molecular machine is truly amazing in how it's able to synthesize ATP. So when the proteins move through this part in green that we call the C ring, there's a special channel just for the protons. When the protons move through it, it's going to spin the C ring, just like the turbine spins in the hydroelectric dam. And when this ring spins, this stalk in the center also spins. And that kinetic motion is used to change the conformation of the purple part, which is the catalytic part responsible for making ATP. And so the machine uses this mechanical energy to take ADP, an inorganic phosphate, and add them together to make ATP. And this is our universal cellular energy currency. And this is the step through this process of oxidative phosphorylation where the cell makes the majority of ATP that it uses to do work. Okay, so I have a very quick video here just to give you an idea. Sometimes we have these simplified diagrams. So this is a little bit more detailed representation of what's happening. The pink region here is the matrix of the mitochondria. Oh, maybe it's not going to play. Oh, there we go. So the pink region is the matrix of the mitochondria. And then the molecular machine, ATP synthase, this is the catalytic part in blue here that's making the ATP molecule. This is the inner mitochondrial membrane. I'll see if the video works. 1 second.
ATP drives
there we go.
activity, driving biochemical activities inside your key building blocks and the key building block and RNA of DNA and RNA.
Okay, so you can see that the machine, so the C ring is rotating, and that's what's allowing that catalytic part to make ATP. ATP is going to drive all the other work that our cells do. So it helps us replicate our genome. It helps proteins transport molecules around. It helps facilitate other chemical reactions that happen in our cells. So I'm going to give you a quick summary of everything we talked about here today. I kind of took a number of themes from different lectures throughout the course, and I've summarized them together. So starting 1.5 billion years ago, an ancient prokaryotic host cell engulfed aerobic bacteria. Through this process of endosymbiosis, the aerobic bacteria became mitochondria, and this developed into a eukaryotic cell. These mitochondria are the metabolic powerhouses of our eukaryotic cell. They supply that cell with ATP, which allows our eukaryotic cell to grow large and do a lot of amazing functions. So when we zoom into this mitochondria and we see what's happening here, we have a series of energy transformations. We go from chemical energy in the food that we eat. So we have a little hamburger here. The fats, the proteins, the carbohydrates are broken down. The carbon-based molecules that they're made out of are going to be oxidized. We breathe out the carbon dioxide through that process. The energy in those molecules are now present in these high energy electrons that arrive. This is my electron transport chain here in the inner mitochondrial membrane. So they travel down the electron transport chain. The energy of those redox reactions, when the electrons are transferred from one acceptor to another, is used to pump protons and make this electrochemical gradient, which is another form of energy. It's like water being held back in a dam. And then when the protons flow through ATP synthase, the energy in that electrochemical gradient is converted into mechanical energy, which is used to synthesize ATP, our universal energy currency. Okay, and then just going back to the larger theme here, so that process of cellular respiration makes ATP our energy currency. It helps then facilitate all the molecular energy transformation so that our cells can do work. And that's the main take home that I wanted to share in this lecture today. And I'm happy to answer any other questions that you have about the topic or the course later on after I think Kara has a bit more to share next.
Yes, thank you so much. That was such an informative sample lecture. I know I took biology in high school, but that was definitely a lot more in depth than what I learned. So it was really cool to kind of take what I had learned and bounce off of that. So thank you so much for that. I'm just going to share my screen with you all now. So if you haven't come to any of our sample lectures yet, I just do a quick presentation at the end talking about how to get into this program. So if this really inspired you today and this is something that you'd really like to get into, biological sciences, this is kind of how you're going to find your pathway to the University of Calgary. So this is just a little bit about the University of Calgary. So it's UCalgary at a glance. These are some statistics that you might see kind of around the web. You might see some from other schools, but these ones are ours. So we were founded in 1966. We have about 28,000 undergraduate students, 5 campuses, and then 14 faculties. 10 of those are undergraduate faculties. So those are faculties that you can enter into straight out of high school. And that's all done in under 100 years, which is pretty impressive when you think about how young the university is. We're also Canada's entrepreneurial university. We have about $504 million in sponsored research funding, and then a 94% after graduation employment rate. And my favorite statistic of all is the 95% undergraduate retention rate. So the undergraduate retention rate speaks to students who come to us for their first year and will follow through their entire degree with us 95% of the time. So that just speaks to student life satisfaction on campus, satisfaction with classes, with professors, with the content that they're learning. And as a student myself, I can definitely say that that is very, very true. So you can see here, this is just a little bit about our admissions information. So the general information that we're looking for is 5 faculty-specific classes, and the admission is competitive, which means we're going to be looking at your averages from those classes competitively. Those ones are all going to be grade 12 high school subjects. So there are some programs that have additional supplemental applications. So if we're thinking health sciences, they have an additional written piece that you're going to submit to us. For fine arts, dance, and music, they have an audition. And then for fine arts, visual studies, they have a portfolio that you have to submit. So just some additional pieces for those guys. But there are all the other programs are just going to be based off those 5 faculty specific classes. And then all students must also demonstrate English language proficiency. So you can see here, these are just the specifics for biological sciences. So those 5 classes are going to be English. So it's an English class. You're going to see this across the board in all of our programs. You're required to have that English class. For Alberta students, it's English 30-1. For other students, it's just that kind of grade 12 level English that you can take. So Alberta Mathematics 30-1 or equivalent. So that's for anybody out of province or out of country. We're looking at that pre-calculus class. And then 2 of biology, calculus, chemistry, physics, or computer science. We'll take a combination of those 2 classes, and then an approved course or option. That approved course or option can be a social studies class, it can be an additional science class, it can be an option, so something like physical education, fine arts classes, those kinds of things can be used as that improved course or option as that 5th class. That's included in that high 80s average that we're going to be looking for. So the high 80s average is just an average of those 5 classes. Not every single class needs to be in the high 80s, but we're looking for it to be an overall average of those. Then these are just some important dates that we have. So for any students who've received an offer for fall 2024, you're probably familiar with a few of the dates on the chart here. The one that's coming up in the next couple of days is the decision deadline, which is May 1st. So if you haven't accepted your offer and you haven't paid your deposit yet, May 1st is the deadline to do that. For any students interested in fall 2025, we're going to be looking at August 1st is going to be the application opening for international students. And then October 1st, it's going to be open for domestic students. And for everybody, it closes on March 1st. So you kind of have that timeline to submit your application, get your transcripts into us, and then there's some additional dates on the screen here. But trust me, you will be reminded of these, so don't worry about forgetting about them. And then these are just some ways that you can connect with us. So if you'd like to speak to a region-specific recruiter, we have them all around the world. You can connect with them at the QR code here on the left-hand side of the screen. And then you can also download our viewbook just at the center QR code there. So that's going to be the most updated viewbook that we have. And then on the right-hand side of the screen here, there's just different ways to connect with us. So the top one is a future student's email address. If you don't know who to reach out to, just start with that future student's email and we'll point you in the direction that you need to go. We also have our Facebook, so you're going to actually see that we've live streamed this webinar to our Facebook channel tonight. We have the Choose UCalgary Instagram account, the Choose UCalgary Pinterest, and then the Choose UCalgary podcast. So if you like listening to my voice, you can go back and listen to season 8 of the Choose UCalgary podcast. And then all of our sample lectures are going to be posted on the Choose UCalgary YouTube account. So if you haven't already gone to check out some of the previous ones, they should all be uploaded. The last ones were uploaded today. So you can go and check those ones out. And then these ones will be uploaded by the end of the week. So you can go and rewatch them if you missed anything kind of throughout the presentation. And that's all I have for you guys tonight. So like Brianne said, if you have any questions, please feel free to put them in the chat. I'm going to leave it open for about 2 to 3 minutes to see if we have any questions come through. If you don't have any questions, thank you for coming tonight. I really appreciate everyone coming out. it's always really fun to kind of get a glance at what university can be like. I know that was something that I was really interested in when I was first getting into from high school into university. I wasn't really sure what a lecture would look like. So these are really, really helpful. And I just wanted to say thank you again, Brianne, for coming on and giving a lecture tonight. It was really, really appreciated. I'll just open the chat to see if anybody has any questions. You can also utilize the Q&A function down at the bottom. So the slideshows for the lectures, they're just going to be in the recorded format. So we're not going to be posting the slides afterwards. They're just going to be on that YouTube channel. So that Choose UCalgary YouTube channel is where you'll find all of the recorded lectures. Yeah, you would just want to go to the YouTube channel there. Here, let me find the link for you all just so you can grab it. The other ones should be uploaded here soon. They just are taking a little bit because they're a little bit longer, but it's nice because you'll be able to see all of them.
Let's see,
I think that one might
question
be
in
for
the
you,
chat.
Brianne, if you want to answer it.
Yeah. So what types of advice would you give us for success in biology and science type courses? I can speak to biology. So I teach first year biology and second year biochemistry. I think really, especially in the fall semester of first year, it's really thinking about time management, and planning your own schedule for studying and reviewing that leads to success in that type of course. So I know that a lot of students have a lot going on, especially if they're moving or such in that first year. So really thinking about setting aside time for when you're going to study, when you want to review, trying to meet some students in the course, especially in the lab section. So that's your chance. That's your small group experience where you'll have a lab partner, exchange contact information, get to know the people in your lab section, so that you can study together, kind of trade those study techniques. And I know that the university as well has a lot of great resources and tools for it's a Student Center for Success. So that is a good place to check out early on for extra support if you feel that you need it. They have a lot of useful information about time management, how to study for exams. There's specific things, how do you answer multiple choice questions? How do you preparing a written response? So that can be very helpful as well, just to find those resources and know what they are early on when you start in university.
Yeah, I know when I first started in my first year, it was all about time management and just kind of getting to know a little bit more about how to properly answer multiple choice, how to do written responses, making sure that you're going to class, going to lectures, going to tutorials, everything that's available to you, you want to make sure that you go to it. So definitely taking advantage of that.
Yeah, and just know that your instructor is there to support you, and same with the TA who will be leading your lab section. We're there. We want you to do well. We want you to succeed. So come to us with questions, and we're happy to help as well.
Oh, it looks like we've got one just in the Q&A section.
So during the course, are we going to discuss the metabolism of only eukaryotes or bacteria too? So we do actually discuss different types of metabolism, and we cover bacteria as well as eukaryotic cells. So we go into a lot more detail. We also talk a little bit about the metabolic process in organisms that don't actually require molecular oxygen, so they have a different electron acceptor. And so you see like those diverse kinds of metabolism in bacteria. So we go into a little bit of detail about that as well. There was a question about tutorials as well, scheduled throughout the year. So some courses will have labs scheduled, other courses have tutorials, and in most cases, at least in science courses, those tutorials are smaller group sessions where they could be led by a TA, so a graduate student, and rather than doing a hands-on lab activity, you might do more of like an in-depth discussion or problem-based learning type activity.
And then it looks like we just have one more in the Q&A box down here. So what are some examples of labs that we could apply this topic?
Right. So I don't know if we have a lab on this particular topic, but you do look at enzyme kinetics in the labs in biology 241. So that would be looking at enzyme that's going to catalyze a particular chemical reaction, and you'll learn how to use the spectrophotometers in the lab to measure products that are formed over time during that chemical reaction. And then you can begin to quantify some aspects of the reaction. So you can look at the maximum velocity of the enzyme catalyzed reaction. And you can look at what happens if you add an inhibitory molecule to that enzyme. How does that affect the maximum velocity of that reaction? So that's one example of a lab that you would do in the course that's focusing more on molecules, biomolecules like proteins that can act as enzymes. So there's a question about prerequisites for biology 241. So I know that you do need, if you're studying in Alberta, you need biology 30, chemistry 30, I believe. I'm not sure how, if there's international students coming, I'm not sure how those prerequisites are. If you're coming from different parts or different provinces or different countries, but I'm sure admissions would be able to help answer that question.
Yes, I'm just putting a link in the chat right now. So it's the university catalog, and that can be really helpful when looking for those prerequisites. So the big thing is you'll just want to go into the search function and put in the course code, and then it'll pull up all of the information about the course as well as the prerequisite classes that you're going to need to get into that. So as you get farther along in your degree, you're going to find that some classes have, some first year classes or 200 level classes that are required as a prerequisite before you can get into it. And this catalog is going to be really helpful to determine what those classes are and plan your schedule out. So you're just going to want to look into this link that I just posted, Abby. And then I think we might end it for tonight. But thank you so much for coming on, Brianne. It was really great, hearing you speak about the mitochondria. I know it's definitely something that's very interesting to think that small in the world, but it's very, very cool. So thank you so much.
Thank you for hosting, Kara, and thank you everyone for joining us today. And I hope I will see some of you in the fall.
Yes, definitely. Awesome. Thank you so much. Have a great night, everybody.